Hello, this is Mr. Philippek, and this next video in this DNA series is going to talk about this question, so now what? What do we do now that we know the structure of DNA, we know how it makes a copy of itself, so what's kind of the next logical thing that we do? Well, I think it's important to start off that we understand that DNA codes for the making of proteins. And everything in our body is made of proteins, whether it be skin, bones, hair, fingernails, nose hairs, doesn't matter. Other types of proteins in our body are enzymes. And remember, enzymes basically speed up chemical reactions in our bodies to allow those millions of chemical reactions that happen every second happen at a much quicker rate. Now, we also remember way back in biochemistry that we talked about the building blocks of proteins, which are amino acids, okay? And so amino acids are important because they are going to be the building blocks of what's going to determine which proteins are made. Because remember, in our earlier video, we said maybe we had the amino acid sequence that spelled the word Earth. And again, these are fictitious amino acids. So Earth is definitely going to make one type of protein. Well, if we switch just some words around here, we're going to get the word heart. Using the same exact amino acids here, just changing the sequence, I think we all both agree that Earth is much different than heart. And the same thing goes with proteins, that depending upon the sequence of these amino acids is going to depend upon which protein is going to be made by the cell. And so now what we have to do is figure out how do we go from DNA, which is in the nucleus of the cell, all the way out to this protein, which happens in the cytoplasm. Well, one thing we're going to learn really quickly is that DNA is too big and too fat to get out of the nucleus. And so what we have to do is go through a couple of other processes. One is called transcription, and the other one is called translation, and those will be covered in other videos in the series. But the idea is, how are we going to go from this recipe book inside the nucleus all the way out to the cytoplasm to make a protein that can be used by a cell? Well, we have to use this new molecule called RNA. If we remember that DNA stands for deoxyribonucleic acid. That's how we get DNA. Now, RNA stands for ribonucleic acid. And so there are some definite differences and some similarities and some contrasting points to RNA and DNA. First and foremost, remember that DNA is double-stranded. Remember, we called it a double helix. And RNA is single-stranded, all right, which means it's smaller. So if DNA is double-stranded, which means it has, two strands, RNA is going to be single-stranded, look just like that, okay? The other point here we have to understand is the sugar. The sugar in DNA, remember, is deoxyribose, but the sugar in RNA is ribose. And if you remember that the reason why we call it deoxyribose in DNA is because it's missing that oxygen group on carbon number 2. Well, obviously in RNA, that oxygen comes back, and we have that normal shape of that 5-carbon sugar, ribose, right? And so right off here, we're going to have that OH group right here, which is how we end up with ribose. The other thing we need to look at here is the difference in the nitrogen bases. Okay, remember with DNA, we always have A pairs with T, C pairs with G. But if you notice right here, we have a difference in nitrogen bases. In DNA, we have thymine. But in RNA, we have this new molecule called uracil. So it's going to kind of revamp some of the base pairing relationships. So like in DNA, we have A goes with T, C goes with G. When we pair up things with RNA, whenever we have to convert things to RNA, A is now going to pair with U. C is still going to go with G. And then I like to add a third one here. Matter of fact, I like to put an arrow here as A goes to U, C goes with G. And then I always like to add in that T in a one-way relationship will pair up with A. And we'll kind of have some practice here in the next video when we talk about transcription. But this slide, we pretty much just want to make sure we understand the differences between RNA and DNA. So what is the job of RNA? Well, its main job is to copy DNA. And so for the whole purpose that we need to get the DNA from the nucleus out into the cytoplasm. And there are three basic types of RNA. We have something called messenger RNA, which is going to carry a message. We have another one called ribosomal RNA, which is going to act like a factory to make the protein. And then the third one is called transfer RNA. And its main job is going to be what we're going to call a supplier. So let's talk about each of these one at a time. So here we have a picture of somebody delivering a package, or a matter of fact, a message. It could be something from UPS, FedEx, whatever, DHL. Basically, the idea of messenger RNA is to take this message from the nucleus and deliver it all the way to the cytoplasm. And so what happens here is that DNA, is double-stranded here. And what's going to happen here is we're going to have this little thing called messenger RNA. After DNA unzips, this single-stranded thing of messenger RNA is going to be created. It's this message which is going to be sent out to the cytoplasm, which is eventually going to be the recipe for the protein. So it's like when you send a text message or a Facebook post or something, you're sending out a message. You're basically transcribing what's in your brain and putting it on a computer. Same thing with DNA. Well, that message is going to be sent to the ribosomal RNA, or what we call rRNA. And I always like to bring up the idea of Chuck E. Cheese, because ribosomes, remember, are found in the cytoplasm. And what happens here is that ribosomal RNA, which is right here, these two big red subunits, kind of form this double structure. And what happens here is messenger RNA is fed in right into that center, which we call the factory. And much like if you've ever been to any type of place where you can play skee ball and get a bunch of tickets, that message is fed through. And as it's fed through, just like when you feed your tickets into the machine counts up how many tickets you got. Well, what ribosomal RNA does is it begins to read the sequence of messenger RNA, and then begins to solicit the help of the next form of RNA, transfer RNA. And we call transfer RNA the supplier. And what transfer RNA does is it basically brings the amino acids to ribosomal RNA. And the reason why we call it transfer RNA or tRNA is the molecule looks like a T. And what we're going to learn here is that there's going to be a sequence of 3 letters right here. And this sequence of letters is going to match exactly what is on, or be complementary to what's on messenger RNA. And on the other end of transfer RNA right here, we have that amino acid. And we'll talk about the 20 different types of amino acids in the translation video. But the idea is that transfer RNA is going to ensure that the amino acid sequence is brought in exactly in the right sequence. So again, if we wanted to spell the protein Earth, transfer RNA is going to ensure, by using a series of codes, that we have the exact amino acids in the correct sequence. So the E comes first, followed by the A, followed by the R, T, H, and so on. All right, and so remember, transfer RNA is a supplier. Ribosomal RNA is kind of the factory where the protein is going to be assembled, and then messenger RNA carries a recipe. And so here we have some review questions. And at this point, I'd like you to, on a separate sheet of paper, pause the video and try to answer these questions. And just tell me what type of RNA is going to do the job that's described in each of the questions here. And so why don't you pause the video and then start it up again, and we'll go over the answers. Okay, so I hope you had a chance to answer these questions. And so the first question says, how does information from DNA get to the cytoplasm? That would be through our good friend messenger RNA. How does the zipper get unzipped in DNA replication? Well, hopefully we know it's an enzyme. And that bell's telling me my video is getting too long. But it's an enzyme. And the enzyme's actual name is called helicase. Now, who does the work in getting the amino acid to the worker? We call that tRNA. And what is the name of the worker of the factory? ribosomal RNA. Well, I hope this gave you a great background on what RNA is, and thanks for listening.